So, messenger RNA. After we make the messenger RNA, after the transcription, what happened? The messenger RNA that we made from transcription is not finalized yet. We need to have some processing to it or modification to it. The messenger RNA that came right out from transcription, we call that primary messenger RNA, or we call it precursor messenger RNA. Many textbooks call it pre-mRNA. So the pre-mRNA or precursor messenger RNA, they have they contains intron and exon. What does intron and exon means? Introns, they are the so-called intragene. Well, let's talk about exon first. Exon, they are the sequence of the gene that can be expressed as protein. So what you can memorize that they can be expressed. EX, go with EX. In other words, exon, you can call it the useful sequence. How about intron? Intron, they are the intragene that are getting removed. Or usually I tell students that intron, they are the so-called the junk sequence that are inserted in between the exon. So intron, they are inserted between exon. So you have the IN goes with IN. As a matter of fact, intron, they are not really junk sequence. Intron, after we remove introns from the messenger RNA or the pre-RNA, it shouldn't be primary, it should be pre-RNA. After we remove the intron from the pre-RNA, then what happened? The intron can be recycled. We can use intron to stop the process of translation. Yes, the process of translation, which is the next stage. So intron is not really junk sequence. They can use it to stop the process of translation in case any mistake happened during transcription. And the processing of messenger RNA, so the first thing is to remove the intron. After we remove the intron, so the entire messenger RNA will contain only the exon. And then we also put something to the end. We put a guanine cap at one end and poly A tail to the 3' end. So we put something at the end of the messenger RNA to have it to stabilize it. And then some people call it the mature RNA, mature messenger RNA. Is ready to do what? Is ready to move from the nucleus to outside. When you go through the nucleus, nuclear membrane, you have to go through the nuclear pore. So this is a schematic diagram to describe the messenger RNA processing. You can see that, okay, the transcription occurs from DNA into pre-messenger RNA. The pre-messenger RNA, it contains an exon and intron. And then you put a cap here, poly A tail and guanine cap. And then there's something called a spliceosome. The spliceosome here, it will remove the intron. Spliceosome here will remove the intron, and then we call it splicing. It's like cutting it out. And after this, the mature messenger RNA will contain exon only. And the cap, the guanine cap and the poly A tail, and then it is now ready to come out from the nucleus through the nuclear pore to go to the cytoplasm. When you go to the cytoplasm, then you can start the translation process. So this is a very simple question. Try to think about this. What is the most important bond in physiology or in a cell, in a human cell? I remember asking the same question in the first exam, but it's like a review. I'll tell you the answer in the next video. So translation. Translation is the second process by which gene expression leads to protein synthesis. So now you got the messenger RNA. How do we make a protein out of it? Translation requires several enzymes and different RNA molecules. It requires these three RNA molecules: messenger RNA, transfer RNA, and ribosomal RNA. So in order to start understanding how a protein is formed, we need to know that there is a genetic code. The triplet code, we call it the codon. Okay? Codon is, remember, codon is found on the messenger RNA. Messenger RNA contains codon. There are 64 different messenger RNA codon. Around 61 of them, about 61 of them, they code for amino acid. Therefore, there are some redundant code. Remember, in human being, in human body, we require 20 amino acid. We require 20 amino acid. And then we have some redundant code. That means some amino acid, they have some amino, they have more than one codon to code for the same amino acid. Why is it? In order to prevent the mutation. When mutation occur, at least they still code for the same amino acid. So it's like a protective mechanism. The repeated redundant code. So that means more than one triplet codon can code for the same amino acid. It helps to protect against the mutation. And then there are three codon. We call them stop codon because they do not code for any amino acid, but they tell the ribosome to stop making a polypeptide. So the three codon that does not code for any amino acid, but it will stop the entire process of translation. We call that stop codon. And the last point is very important. All living organisms, including tree, including plants, including the cockroach in your kitchen, including the flea on your dogs, including the E. coli in your large intestine. We all use the same genetic code. So this is what we call the genetic code. So this one, we call it, how do we read this table? First of all, we read from the first base. First base is here. For example, this is A. So the first triplet code, the first codon is start with A. And then the second base is horizontal. Let's take U. AU. What will be the third one? The third one, AU. That means here. And this one is called AUG, AU, and then G. AUG, we call it start codon. Start codon, AUG, always code for methionine, this amino acid. So this is something that I asked you to memorize. Start codon. it is called AUG, and AUG always code for methionine. This is something that is better for you to memorize it. And then we have this 3-stop codon. UAA, you can see that U is here, A, second is A, the third one is also A. We call it stop codon because it doesn't code for any amino acid, but it stops all the entire translation process. And UAG, UGA, and UAG. So all these three are the stop codon. And then in the previous slide, we talked about the redundant code. Let's take a look. You can see that all these here, they are proline. But they only differ from the last codon, UCAG. And they all code for proline. The redundant codon, that means CCU, CCC, CCA, and CCG, they all code for the same amino acid. The only difference is the last codon because it helps to protect against the mutation.